Welcome to Thursday. While all of your colleagues are joining us, I'm going to go ahead and go over the agenda for the day. We do have a super packed day today. I'm really excited about all of the presentations that we're going to hear. And I wanted to send a special shout out to our lightning talks this morning, Sissy and Guess. They were really super. So if you didn't get a chance to watch them, please do catch up and we'll try to get those videos to you. They were great. Okay, let me share my screen for just a quick moment and go over the agenda. So today is Thursday. Welcome to Thursday. We've had our lightning talks already this morning. We're going to kick it off in just a few moments after Dave does his organizer corner chat with our project presentation 5. We'll take a little lunch break, and then we're going to hear 2 different types of talk than what we've had in the conference so far. We have our first annual FYIM alum presentation, which I'm really excited about, this being kind of the inaugural chat in this conference. And then we're going to hear from one of our co-PIs about the website that's being built for networking. So that will be coming up after lunch today. We will then move into project presentation 6, take a quick break, and then we're going to hear from one of our NSF program officers who's been with us this week. He'll give a little funding talk, and then we'll have some time for Q&A with him. So I highly encourage you to come to that as well. And then we're going to have a few more lightning talks this afternoon, which I really encourage you to attend. They've been really fun and interesting so far. So that is the agenda. Just as a reminder for questions throughout the event, throughout all of these different talks, every page has been set up with questions at the bottom. So just a reminder, when you come through and you're like, yay, project presentation 5, I can't wait to ask all my questions. You just need to click this little plus button, type in your text, and then hit that paper airplane, and they will be able to see your questions. I'm really excited to say that we've kind of been working our way back in time, and I do think most questions have been answered at this point. So when they're not answered live in front of the room, we are following up with presenters to ask if they can please answer any questions that were not addressed. So if you asked a question, feel free to go back and see if that was answered. It could have been answered in the room if it doesn't have a comment on there. And you can follow up with Jocelyn, Malachi, or I if you're like, wait, no, my question wasn't answered, and we can go and try and track that down for you if you like. So with that, I think I'm going to go ahead and turn it over to Dave for the Organizer's Corner.
Great. Am I on?
You are on. Okay,
Organizer's Corner. I'm thinking of renaming this to, guess what Elmo's thinking about? Angela DePace's talk. That's right, Angela DePace's talk. So yeah, this is what I've been thinking about since yesterday. 2 things about that talk really struck me. First, a little backstory. A few weeks before the meeting, Angela was communicating with us, and she asked me, would it be all right if she included 2 of her postdocs in the talk? And I said, you know, of course, you're Angela DePace. So I didn't really know what she had in mind, but I figured she knew what she was doing. And indeed, I think it was a brilliant thing she did. She presented the biological problem, and then she had 2 people talk about a data-driven model, an empirical model, and a theoretical model. And that led, I'm going to share my screen for a minute. The moderator and introducer was Liz Haswell. During the session, she chatted this question: what is the best way to integrate the 2 modeling approaches, data-driven versus theoretical? How do the approaches meet, complement, and synergize? And I think this is a very interesting question. I don't think we even began to answer it. So I think this is something that someone could write a paper or an essay. They could title it How to Reconcile, Integrate, and Synergize Data-Driven and Theoretical Models. They could publish it in the Journal of Reconciling, Integrating, and Synergizing Data-Driven and Theoretical Models. But all kidding aside, I think this is a very important question that I haven't really seen anyone explore. So that was the first thing. The second thing was during the presentation, and I guess it was actually during the question and answer session after the presentation, the presenters kept getting questions along the lines of, why don't you do this? And why don't you ask this? And you could really find out some important things about development down the line. And at a certain point, Angela came back on screen and she said, I want to take this question. And what she said is that she appreciated the curiosity that was driving these questions and the biological interest that was there, the wealth of new information that they could mine. But this isn't what this project was about. What she said, these questions weren't really in line with the philosophy of this project. And she elaborated that a little, but I didn't feel it was 100% clear. So later, when we were back in the green room, I asked her what she meant by philosophy. I said, did you essentially mean you're trying to answer this particular question? And that question was largely, how do these transcription factors interact and what roles do they play within this cycle of transcription? And she said, yeah, it's really, I was just saying, those questions are beyond what we're trying to model. And I said, well, why did you use the word philosophy then? That's a little confusing. And then she elaborated what she meant by that was the philosophy was including not just the modeling, but the methods of making the measurements, the experiments. And they really wanted to stop with a very high level readout of transcription. People were wondering about particular transcripts. They're not interested in that. They just want, at this point, to know when are the nuclei transcribing actively, when are they not. And so she was including that as part of the philosophy of the experiment. So I think this is interesting because it made me realize that there's a common danger that I think modelers, biologists and their collaborating modelers face, which is once they have a model, they start going down the slippery slope of, oh, well, we could do this, let's try this, let's tweak that, let's pull here and see what happens. And then you can easily lose sight of the initial question. So I think what Angela was telling us is it's always important to keep that question in mind and keep the modeling on track. I kind of think of Frankenstein, and how Dr. Frankenstein unleashed this guy on the countryside. You don't want your model roaming the countryside. Keep it in the lab. That's what I have to say about that. And that's really it. That's the things I wanted to talk about in this little feature this morning. And I think at this point I can pass it back to our introducer, or I guess it's Donalyn or Jocelyn, to introduce our first talk.
It is me. Thank you very much, Dave. So it is my great pleasure to introduce Project Presentation 5 by Stephanie Wu and Tom Wyatt. Our invited experts will be Andrew Mugler, Eric Dietz, and Leonard Harris, who will be chipping in at the end. First, you're going to hear from Stephanie, and then Tom will take over. Their project is titled Endodermal Cells Use Contact Inhibition of Locomotion as a Cell Spacing and Dispersal Mechanism. Maybe, yeah, I saw Elizabeth give me the head nod, so awesome. Okay, so with that, Stephanie, the floor is yours. We'll make sure that your audio and the images look okay.
Okay, great. Thank you so much for that introduction. Let me get the slideshow started. Okay.
Looks great, thanks.
Great. Yeah, so I'm excited to tell you guys about a project that we've been working on in the lab and that we've recently started collaborating with Tom on to introduce some modeling aspects to it. We are looking at so this is a zebrafish embryo. This is the model system that my lab works with. In the movie that you're watching, all of the nuclei of this embryo are labeled in red. And the tissue that we are interested in, which is called the endoderm, is labeled in green. And so what I'm going to tell you about today is a behavior called contact inhibition of locomotion that we think is important for the morphogenesis, or for the shaping of this particular tissue. And before I get too far into the presentation, I do want to acknowledge that this project is primarily the work of a lab technician, Jessalyn LaBelle. So for those of you who don't have a background in developmental biology, we work on an embryonic tissue called the endoderm. It's one of the 3 primary germ layers, so one of the first tissues that arises during development. So along with, so there's the endoderm, the mesoderm, and the ectoderm are considered the 3 primary germ layers. The endoderm in particular is the tissue that will eventually give rise to the epithelial lining of your gastrointestinal and your respiratory system. So it will contribute to the epithelial tissues of all of these organs labeled down here. But if we look back in developmental time to when these embryonic germ layers are first formed, you'll see that they have, in all animals, they have this very stereotyped arrangement where the endoderm is located in the inner part of the embryo, so it forms the innermost layer of the early embryo. The ectoderm forms the surface, and then you have the mesoderm sandwiched in the middle. So this arrangement of the endoderm being placed in the inside of the embryo means that when the endoderm is formed, those endodermal cells need to find their way into that inner layer of the embryo. And so different animals have evolved different mechanisms to place the endoderm on the inside. So some organisms like Drosophila and amphibians like Xenopus, they use those strategies involve movement of the entire tissue. Tissue folding or invagination for Drosophila, or tissue spreading, or what's called involution in amphibians. However, other organisms like the zebrafish that we work on, or also avians and mammals, they take a slightly different approach to placing the endodermal cells on the inside. So in these organisms, the endodermal cells actually sort of delaminate and actively migrate as individual cells and move their way towards that inner part of the embryo. And so for us, this is really interesting because it means, so again, remember that the endoderm will eventually give rise to the epithelial lining of all of these organs. And so that means that if you, in an organism where the endoderm takes this approach of delaminating and actively migrating to reach the inner part, it means that at some point, those actively migrating cells need to transition back to a more coherent, stable epithelial tissue. So we really like looking at endodermal development as a model of what are called mesenchymal to epithelial transitions. So this transition from a single cell migratory state to a more stable, coherent epithelial state. And these types of transitions, you see them over and over again in different developmental and disease contexts. And so we think that in the zebrafish in particular, the development of the endoderm is a really good model for looking at these mesenchymal to epithelial transition decision-making process. So these are just some cartoons giving you a quick overview of what endodermal development looks like in the zebrafish. Obviously, this is a highly schematized diagram of an early zebrafish embryo. So again, the endoderm is one of the 3 primary germ layers, so we're looking at very early development. And so at this stage of development, the zebrafish embryo basically just looks like a sphere. And the embryo itself, you can think of as a little ski cap sitting on top of a giant ball of yolk. And the endodermal cells will be specified in this band of cells that lie right at the margin between the embryo proper and the yolk, so sort of the edge of the ski cap. Soon after specification, those endodermal cells will undergo that ingression event. So what I was saying before, they delaminate and they start actively migrating to flip underneath their ectodermal and mesodermal neighbors to form that inner layer of the embryo. But during this internalization process, the endodermal cells actually lose contact with each other and they disperse throughout that inner surface of the embryo. And so from previous work, we know that this dispersal process is characterized by directionally random migration. So they have very low directional persistence at this stage. And as we'll see later in this talk, they have very little to no contact with each other. So they form this incoherent salt and pepper-like distribution of cells. However, at some point later, this dispersal behavior ceases and the cells start to converge together on one side of the embryo, on the dorsal side of the embryo. So this convergence process is characterized by highly directional migration. So they all gather on one side of the embryo, and we can actually see them reach out and make contact and increase intercellular adhesion with each other. So one of the advantages of working with zebrafish is we can observe all of this in real time because the embryo is very amenable to live imaging, live microscopy techniques. And we have a whole library of fluorescent transgenic lines that help us visualize these processes. So this is one particular transgenic line that we use a lot in the lab. This is using an endodermal SOX17 promoter, just driving expression of GFP. So before I start this movie, you'll notice there's this very brightly fluorescent group of cells down here. These are actually non-endodermal. They're called dorsal forerunner cells. They're also interesting in their own right, but they're not the focus of today's talk. So what I want you to focus on are the slightly dimmer endodermal cells out here. And you'll see they're already sort of dispersed. And as the movie plays, I hope you can appreciate that, one, their migration is very dynamic, and two, towards the end of the movie, you'll start to see them converge. In this case, they're converging towards the middle of our field of view here. So I'll let that play one more time before moving on. So what we're interested in is that decision-making point between dispersal and convergence. And so we started making high-resolution time-lapse movies of embryos during this critical transition period. So this is a different transgenic line from the slide before. So in this line, we're using this endodermal SOX17 promoter to express a fluorescent actin binding protein. So this transgenic line will give us information about actin dynamics in these endodermal cells. So if we look at about 8 hours post-fertilization, so during the phase when cells are still sort of in that dispersal mode, we see that cells make transient contacts with each other, but they're very short-lived. If we look about an hour later, so this is the same embryo, just 1 hour later, again, we see cells occasionally reaching out, and they seem to hold on for just a little longer, but they still eventually pull away. And now if we look another hour later, so 10 hours post-fertilization, you can see that these two cells are already flush with each other. And you'll see a third cell come in and join them. And so that cell will, and you can see that these contacts are more permanent. So they seem to really stick together. So I like to call this the romantic comedy model of, of cell adhesion. So first you avoid each other and then you love each other. So we can, let me just stop those movies. So we can quantify these changes in contact duration over time. And we see that at these later stages, we do see a significant increase in the amount of time these cells spend in contact with each other. We can also correlate this increase in intercellular adhesion with changes in expression of adhesion molecules like E-cadherin. But what I will talk about today is we think that there's an additional mechanism involved that's driving these dispersal versus convergence behaviors. And that behavior is contact inhibition of locomotion. So what we observed is that cells in endodermal cells in the early dispersal phases of development appear to actively avoid each other. I can show you that in this movie here. So if you just follow any two cells in this movie, anytime that it bumps into another cell or collides with another cell, the two cells appear to push away from each other. So again, using our actin reporter line, which here is pseudo-colored based on actin content. So warmer colors indicate more actin content. We can see that these collision events are correlated with a decrease in polymerized actin content, rapidly followed by membrane retraction at the site of contact. And you can see that also correlates with the appearance of new actin polymerization on contact-free edges. And it's this new actin polymerization that appears to allow the cells to push away from each other. So that's also diagrammed down here. And so these observations are very much in line with this behavior called contact inhibition of locomotion, or CIL, which again, the key characteristics here are actin depolymerization and membrane retraction at the site of contact, followed by a new actin polymerization on a different edge of the cell that allows the cell to move off in a different direction. So CIL has been described and observed in lots of different other cell types and other contexts. And so in other cell types, there have been several different cell surface molecules that have been identified to mediate this CIL process. But of all of the different contact inhibition of locomotion receptors that have been identified, they all almost universally converge on a signaling molecule called Rho-A. So Rho-A is a member of the Rho family of GTPases, and Rho-A has a very well-characterized role in triggering membrane retraction through activation of myosin activity, as well as regulating actin dynamics. So it's really well positioned to signal downstream from some kind of contact receptor to initiate these processes that are key to this CIL behavior. So then there are two questions that we're interested currently. So one is similar to what has been described in other cell types. does endodermal CIL also require Rho-A activity? And secondly, what are the receptors that allow endodermal cells to recognize and push away from each other? So taking the first question, so to determine if endodermal cells require Rho-A for CIL, we injected in zebrafish embryos a dominant negative version of Rho-A. So this will block Rho-A activity. And what we saw is now when cells contact each other, rather than pushing away, they just slide right past each other. We can quantify that if we look at the migration angle after a collision event. So the way we did this was we normalized the incoming migration angle to 0, and then looked at the migration, the direction of migration sometime after that collision event. And we found that cells expressing dominant negative Rho-A did not change their migration angle as much as control cells. So we can summarize that here. So if you don't have active Rho-A or functional Rho-A, when you collide with another cell, you just keep going in the same direction you were going in. Whereas in a normal endodermal cell, when they undergo collision events, that collision events will trigger them to change their migration direction. So this was really interesting because so this implies that if we take away Rho-A activity, if we express this dominant negative Rho-A construct, we can get rid of this CIL behavior. So that allowed us to ask, well, then what happens to the endodermal tissue as a whole when we take away CIL activity from the endodermal cells? And so we looked at embryos when the endodermal cells are maximally dispersed throughout that inner surface of the embryo. And we found that when we expressed dominant negative Rho-A, so when we took away CIL, we found that the endodermal cells were irregularly spaced. So normally these cells, not only are they maximally dispersed, but they have this very even, regular spacing between themselves. Whereas in embryos expressing dominant negative Rho-A, we saw irregular clusters of endodermal cells interspersed with large gaps in the endodermal tissue. And again, we can sort of quantify this if we measure the nearest neighbor distances between endodermal cells. And so we found that in embryos expressing dominant negative Rho-A, there was a much larger distribution in those nearest neighbor distances, which fits with there being sort of irregular spacing between these cells. So we've established that Rho-A indeed is probably involved in this CIL process in the endoderm. So then next we wanted to know, how are the endodermal cells even recognizing each other to push away from each other? So we had done some transcriptome profiling by RNA sequencing, and we came up with a list of candidate receptors that are highly expressed during these stages when these endodermal cells are undergoing CIL, but then become downregulated as they converge and differentiate into this more epithelial sheet. Importantly, because this is an example of homotypic CIL, so it's two cells of the same cell type, we reasoned that the ligands for these receptors must also be co-expressed by endodermal cells. So we further narrowed our candidate list down based on those where their ligands are also expressed in endodermal cells. And so one candidate that we're really focused on is the FA2 receptor. So F receptors and their ligands ephrins have a very well characterized and well described role in playing in initiating repulsive interactions between cells, especially in axon guidance and in boundary formation. And so that has been our main candidate so far. So we're currently using CRISPR to knock out both the receptor and the ligand in endodermal cells. But in the meantime, while those mutant lines are getting established, we decided to do some preliminary experiments using a pharmacological inhibitor of FA2 receptors called dasatinib. And so when we treated embryos with dasatinib, we found that the distribution of endodermal cells, again, appeared irregular, similar to what we saw with expression of double-negative Rho A. So we saw irregular clusters of endodermal cells. So putting this all together, what we think is going on is that these endodermal cells are using CIL or self-avoidance through CIL as some kind of positioning or spacing mechanism. And a similar type of mechanism is known to be used in sensory organs and by neurons. So we see self-avoidance as a positioning mechanism in the establishment of retinal mosaics in the eye, in the tiling of sensory dendrites, and also in the positioning of Cajal-Retzia cells in the cortex of the brain. And so we think that these endodermal cells are similarly using self-avoidance through CIL as a spacing mechanism. And so in these sensory organs, it's thought that this type of spacing mechanism allows the cells to maximize coverage while minimizing overlap. So if these endodermal cells are similarly using CIL as a way to maximize coverage while minimizing overlap, there's a couple of questions that we're curious to answer that we think modeling might help. So the first one is, why is that maximum coverage, why would that be so important for endodermal development? And secondly, is CIL, so is this self-avoidance mechanism, is this sufficient to drive these dispersal behaviors to drive that maximum dispersal of the cells? And so, so I'm going to quickly just introduce you to sort of the hypotheses that we have about these questions before handing it off to Tom, where he will explain how we're using modeling to try to address these questions. So the first is, so why would maximum dispersal be so important to endodermal development? So again, it's important to remember that the ultimate fate of these endodermal cells is to form the epithelial lining of the gut and the respiratory epithelium. And so these organ systems span the entire anterior-posterior axis of the body. So we think, so one hypothesis that we have is that dispersal ensures that cells will span, that you place the cells along the entire future anterior-posterior axis, so that they're there already, and so you don't have any future gaps in those epithelial tissues that will arise later. So the second question, so is CIL sufficient to drive this dispersal behavior? So as I showed you before, when we blocked Rho A activity, we saw irregular spacing of the cells, and we interpreted that to mean that if you take away that this spacing regularity is driven by contact inhibition of locomotion, which is dependent on rho A. However, if you're familiar at all with rho GTPase signaling, you know that rho A actually does a lot of different things in the cell. And so it could be that what we're seeing is not necessarily the role of rho A and CIL, but it could be the role of rho A in any of these other processes that it's known to regulate. So we think we can use modeling to get the sufficiency of CIL itself. Is just that enough to explain the behaviors that we see in our endodermal cells? So with that, I will hand it off to Tom, and he'll talk about the modeling aspects of this project.
Okay. Hello, everyone. I will try to share my screen. How is that? You can see me, see my screen and hear. Oh, hang on. No, that doesn't work. Okay. Hopefully you can see my screen and hear me.
It's not the
Yes.
same. But Tom, it's not the same one that you and I showed before that had the picture on it.
There, yeah, I can see there's a problem.
Otherwise, I would say, yeah, your screen looks awesome. Super cool, nice, bright white. Great.
Okay. So then, with an image now?
Yep, that looks
Excellent. Okay, good. Okay, thanks. So, yes, my name's Tom. I'm a biophysics postdoc in the lab of Benoit Saur in Institut Curie in Paris. I'll start by quickly repeating the questions that Stephanie introduced and perhaps restate them slightly from the perspective of the modeling. So that first question that Stephanie introduced, it's what are the best mechanisms to distribute the cells evenly, which really is the question of when you start with your cells aligned across, we could say, the equator of the embryo like that. What's the best strategy for migration to reach a more evenly distributed spacing? And in the modeling, we're going to be looking really at just two ways that cells can change direction, which is firstly the contact inhibition that Stephanie talked about, and the second is non-persistent migration, which is just that cells don't move in straight lines all the time. They randomly change the direction that they migrate in. And that second question is, well, it stems from why do the cells go to all of that effort to cover the entire embryo when very soon after they're going to reaggregate to converge back onto this dorsal midline. So is there a reason we can understand why it could be important for the cells to distribute evenly over the embryo like that? And so that is the question of starting at this later point when cells have distributed, what importance does that distribution have on the final density of cells along that midline? So the work we've started is so far a very basic agent-based model of the embryo, where the thing we're trying to get right is the motion of the cells over the surface of a sphere. So we start with this sphere representing the embryo. It has a radius that is the same as the zebrafish embryo. And we just put a cell on the surface of that embryo, and the cell has the radius which we have measured from the data. And the first thing we want to do is make the cell travel in a straight line over the surface of that sphere. And just as an aside, just to get us thinking about life migrating over the surface of the sphere, like these cells are, I'm going to talk just for 1 minute about straight lines on the surface of spheres, because they're not always entirely intuitive. So if we are planning a trip between Chicago and Paris, we want to go in a straight line. That by definition, a straight line is the shortest distance between 2 points. And if we forget that the Earth is a sphere, we might plan a route which looks like this, where in fact the straightest path, the shortest path, looks more like that. And if we draw it more, the Earth to look more like a sphere, that becomes more obvious, and particularly if we rotate it round like that, and that path, that shortest path, starts to look like a straight line in the 2D sense. So a side over, we go back to our embryos and we can move the cells in a straight line over the surface of the sphere, and we move them with a velocity again that we've measured from the data. But cells rarely move in completely straight lines. As Stephanie told us, they change direction quite a lot. We can say that they have a low persistence length. So their motion is much more like that. How we implement that in the model, we can say that the cells are traveling in a direction defined by this angle rho. And at every step, we could add another small angle chosen randomly to that angle of motion. We choose it randomly from a normal distribution where the standard deviation is given by this formula here. S is the length of the step that you're taking. And P is a parameter of the model, it's the persistence length. And all this equation means is that once you've taken a step which is equal roughly to the size of the persistence length, you will likely have changed direction by a right angle. Yeah, okay, so then we implement contact inhibition in the model. I've drawn the cells bigger here so it's clearer. And that's simple. Whenever we find two cells overlapping, they change their direction to head off in opposite directions. And what's not quite so clear is how we implement the case, in fact, without contact inhibition. So at the moment, we have the cells moving towards each other. Cells aren't allowed to pass directly over each other. So if they're moving in a straight line, then they're stuck because they're in each other's way. And that would be the end of the story if it wasn't for this low persistence length that we've been talking about, where in fact, when cells collide, they're stuck there for a moment or two, but they eventually randomly repolarize and can move off in different directions. The last point in the model to mention is this Epiboly, which is the enveloping cell layer that Stephanie told us about. And we don't find that the endoderm cells can travel in that outside of the enveloping cell layer. So we incorporate that into the model, and we have an Epiboly which is slowly covering the embryo. And if cells reach the edge of that, they will turn around and move in a different direction. So the average simulation looks a bit like this one, where we start with a number of cells that we've got from the embryo. There's cell division, so that we end up with more like 400 cells. And then, so at this point, we want to measure something from the simulations. And the first measure that we've chosen is a measure of dispersal taken from Schilscher et al., where we divide the surface of the sphere into a grid, into segments of equal area. And we know the number of cells in total in the embryo, and we know the number of segments that we've divided it up into. So we know when we divide it up, we know how many segments we would find if they were perfectly evenly. We know how many cells we would find in each segment if the cells were perfectly spread. And we can make a measure of this heterogeneity by counting the difference between the expected and what we find, and summing over all of the segments and then normalizing. So that's what we do. That measure in the perfectly spread case, or the best spread case, would give a very low number close to 0. And in the worst case scenario, where all of the cells have ended on the same spot, the least distributed, it gives a number very close to 1. And this is how it looks like in a simulation. As the cells disperse over the surface of the sphere, this heterogeneity decreases. So now we're in a position to play with the parameters in the model a little. So we look at the difference in the migration of the cells, in their ability to cover the embryo. In the case, firstly, where there's no contact inhibition, and in two cases of a short persistence length in blue and a long persistence length in green. And what we can see straight away is that the green does better to start with. It can, you spread much quicker, but after a while, the low persistence length gradually overtakes it, and it does better on a long time scale. And when we watch the videos, we can understand that quite straightforwardly. So on the left here, we have the cells of low persistence length, and they seem to be migrating quite happily. But they don't, even at the end of this 9 hours here, they don't reach the poles of the embryo. And that's because they're turning around too often. They're slowly diffusing towards the poles, but it's taking longer. Whereas in the case on the right here of the long persistence length, the cells can reach further parts of the embryo faster because they're not turning around. But we have this other problem, which is becoming apparent, that when cells do collide, it takes them longer to separate again because they're trying to move more in a straight line. And you get these aggregations forming, which are really hampering the ability of the cells to cover the entire embryo. If we look at the long time scale, you can see what's happening more at these later times, that the cells, the small persistence length, are managing to cover the entire embryo. Eventually, where in the case of a long persistence length, those aggregates are remaining for much longer time points, and they hamper, as I said, the ability to disperse properly. Now, when we look at the case when we add contact inhibition into the model, we get a much simpler result where, again, with a short persistence length, cells are taking a longer time and a gradual, they're taking a longer time to decrease their heterogeneity compared to this long persistence length where they're doing that very fast. So again, on the left, we have the low persistence length, which looks very similar to the case before, where you don't have contact inhibition. But the case of long persistence length looks much better. The cells are rapidly dispersing and covering the entire embryo. So just to show you that isn't a result of these two particular persistence lengths that we've chosen, we look through a whole range of those parameters, and you see a very clear pattern emerging that the longer the persistence length, the faster you can disperse, particularly in the case where you have contact inhibition. So that gives us our first conclusion, which is that contact inhibition, along with a long persistence length, seems to be the most efficient strategy for dispersal. So now coming to that second question, which was, I told you it was what is the importance of the cell starting distribution for convergence. But actually we're stuck on an earlier problem, which is how do cells in the first place find their way to the midline? So what type of path are they taking to the midline? Or what is their strategy for moving there? And the fact is that at the moment we don't know we don't know exactly what the cells are doing. So we look at a few different strategies and see what the outcomes are. And in the first, we just say that at the point when cells start to converge, they look towards the point in the midline which they are closest to and move towards that point. So a simulation of that will look a bit like this, where cells are dispersing over the surface of the sphere. And at one point, they'll all start moving towards the midline. But this strategy gives a very strange looking distribution, where these points at each pole, at the animal and vegetal, or these north and south poles, you end up with a lot of cells there. And that's kind of obvious when you remember that the dorsal midline is only a half line. So if we play this simulation, all of the cells on the other side of the embryo with this strategy are finding this is their closest point, and they're heading there. So that doesn't seem to be a very likely strategy for the cells to be using. We can imagine another one where cells are moving east-west, so across lines of constant latitude towards the dorsal midline, and that this simulation is showing this exactly that, where cells, you can see they're moving exactly along these lines of constant latitude, and that gives this distribution which may or may not be more reasonable, and we still need to look to the data to know that. But in reality, the likelihood is that neither of those is exactly the strategy. Probably what is happening is that cells are using chemotaxis. There are probably cells along this dorsal midline which are secreting a chemokine, and that is creating some kind of gradient which cells can follow. But for now, in terms of the modeling, this is the problem that we are at. So to summarize, we've found from the modeling, we think that contact inhibition with a long persistence length is going to be the best strategy for cells for dispersal over the embryo, and that we found that during convergence, different strategies for finding that dorsal midline give very different outcomes in the distribution of cell density along the dorsal midline in the end. So we have lots of to-dos. Firstly, I think in the first part of the first question that we've looked at is we now need to look back in the data and try and understand if those models, the models which have given quite a clear answer to what the best strategy is, whether they do actually fit the data in terms of the patterns of migration that we see in the embryo. And then there is this question, of course, of how are cells finding their dorsal midline, because until we know that, we won't be able to get further towards that second question. So with that, I'll say thank you to everyone, in particular Jesselyn in Stephanie's lab, and to the funding, and to everyone for listening. Thank you very much.
Excellent. Thank you so much, Stephanie and Tom. What a wonderful kickstart to our day today. If we could show them a little emoji love and some cheers. Awesome. Thank you. We are going to go ahead and get our expert panel spotlight. A few questions have been coming up on your page. Please do continue to ask those questions, but we are going to turn it over now to Eric, Andrew, and Leonard. The floor is yours.
Awesome. I don't know, who's running this?
I know I'm not.
Okay, I'll do it.
Okay,
so
no,
that was a Leonard, or you? All right, never mind. I'll just take over. So
you go ahead, Eric.
All right, so that was awesome. That was so unbelievably cool. I thought it's a really cool, really cool way of using modeling to kind of explore conceptually what could be, you know, is this an okay answer? Is this sufficient? I thought that was awesome. I'll start by asking sort of a, maybe not a great question, I don't know, but it's something that struck me. And that is that, so in this process of coming back, of converging, there's not just a problem of how they find their way. They also have to shut off this contact inhibition because they're about to go and hug each other. Mm-hmm. And that's something you see in the data, where over time they start out being like, no, I don't like you people. And then they're like, oh, this is awesome, let's get stuck. And so I guess it's sort of two questions. One is, do you know anything molecularly about how that happens? And two, is there a way in the model to change the intensity of the contact inhibition over time? Because that's what you saw as, like, in the 9 hours to 10 hours to 12 hours, the contact inhibition was getting less intense, or the CIL, the whatever, but in the model, it seems like it's sort of a step function where it like they disperse, and then it's like, now everybody come together. And so anyway, that was something that I noticed.
Yeah, no, that is a really great question and observation. So molecularly, we don't, I guess we don't know is the easy answer. So from my movies, it does seem like it's a gradual process, right? So they stick together a little more and a little more and a little more until they don't let go anymore. So interestingly, when I look at them with the actin reporter, so even when they're undergoing that convergence process and they're stuck together, I still see a redistribution of actin to non-contact edges. So at the molecular level, it seems like there's still some part of that. So if we think of, repolarization of actin polymerization as a part of CIL, it seems like that part of it is still functioning for some reason. It's just that the cells don't pull apart.
Maybe they just get stickier because of those
Yeah,
cadherins.
so that's one hypothesis, is that they just get stickier. And I mean, but it could also be that they are so molecularly, we would say they're turning off myosin activity. So they don't pull apart, but they still redistribute the actin. So but in terms of the modeling, I'm not, I don't.
Yeah, that's hard to, I mean, I think there's ways to make the rules like that happen in the modeling. The other thing that I thought of while I was thinking about this is that if they're starting to stick together a little bit, then that seems to happen before they're all the way at the midline. So they may need to develop a kind of collective motion, and perhaps by still being sticky but still having CIL, but they'll probably move slower because there's a bunch of them, and unless they're coordinating their motion fully, if they're all trying to move randomly, but they only have actin in the sides where they're not touching each other, that'll still slow them down because they're kind of they can't talk. They can't be like, hey, but maybe if there's chemotactic signals plus that's enough. Anyway, yeah, I'll stop talking and let other
Actually, so
just to mention on the modeling, you were absolutely right that at the moment it's a step function, and in fact when they're migrating back to that dorsal line in the model, we just turn off contact inhibition and cells head straight there. So, but it's a good question because it's not completely obvious how you tune down contact inhibition, like what that behavior exactly would be. Does it take you some time to change your direction? Maybe you carry on and slowly or gradually change direction, or maybe you immediately change direction, but not by as much, because at the moment they always go in opposite directions, but maybe you don't go so much in opposite directions. But yeah, it's not clear, and we'd have to look very specifically at the data for that, I think, at what happens at those intermediate times.
Yeah, I think you might be able to develop the rules for your little crawling agents based on what the movies look like. So that could be cool, at least to see if those local rules give you the global pattern. You won't understand why those local rules have evolved or how they're implemented molecularly, but at the very least you can say, okay, are these local interactions sufficient to generate the global pattern we see?
Yeah,
be very
exactly. Yeah, that's exactly how we're thinking about it.
Yeah.
Anyway, other experts, Andrew, Leonard, go.
Yeah, so this was really cool work. I just wanted to give my appreciation for it. So I had a question about the dispersal phase and how to get, best get full coverage of the embryo. So when looking at the comparison between with and without contact inhibition of locomotion from your simulations, Tom, it sort of became clear to me that contact inhibition of locomotion does two things. That when cells get together, they not only go off in opposite directions, but maybe more fundamentally, they don't get stuck, right? So contact inhibition of locomotion is a way to avoid getting stuck. And I was wondering whether just avoiding getting stuck is enough. In other words, the simplest way to do this is indeed allow cells to go right through each other, or maybe they're deformable or slimy and they can sort of wiggle their way through each other. And then I was just curious, this may be addressable with the model. Would that alone be enough to successfully get full coverage without these aggregates?
Yeah, I think we need to test that. Definitely. Like, so there's one thing in the model at the moment which is quite obviously different from the data, is that in the data, Stephanie sees that cells do sometimes not pass through each other, but they manage to get past each other without changing their direction significantly. So that's something we have to look at and find some way to implement that in the model, that maybe there's a probability of passing through, or that you can do it through small changes in angle. Another part of the question is, yeah, this is it just about not sticking? Yeah, I'm not sure. There's one point in the embryo that at the start they're all moving in the same direction. All the cells are moving to the north, to the animal pole, and so perhaps also the contact inhibition at the beginning helps randomize these directions. Like, how do cells ever end up going to the vegetal pole? So perhaps there's also a role there. But yeah, it's an interesting thing. We can explore both in the model and experiments, I think.
So I've got a question actually to follow up on what Eric was asking earlier about like the molecular mechanism part of it. So I mean, it just came to my head when Eric asked that question, you guys were answering it. I mean, is it possible that the dispersal, so one of the questions, right, is why are the cells dispersing the way that they are? Is it possible that the molecular mechanism that causes them to converge is connected to that dispersal, right? That once they get far enough away from each other, you know, that the contact is triggering some kind of molecular program, that once they get far enough away from each other, that program turns off and a different program gets activated that then causes them to go back. So there's actually this kind of physical molecular connection.
Yeah, no, that's really interesting. So are you proposing something like the cell is able to somehow count like the number of collisions they're going to go? So if they don't collide very often, that they think, oh, I must be far away from people, and then
Yeah, well, something like, yeah. So one project I'm working on actually has to do with bone cancer cells metastasizing to the bone. And so when cells press up against the bone, in particular, row A gets activated because integrins and receptors bind on the surface that don't when the cells are not pressed up against the bone, for example. So when row A came up on your picture, that kind of triggered something in my head. So clearly when there's like, you know, external pressures on the cells, different molecular programs can get fired. So that's kind of what I was thinking, that when the cells are pushing against each other, it triggers a program that causes them to push away from each other. So then when they're away from each other, a different program might get activated as a result. Yeah.
Yeah, and also, I mean, you know, this is an embryo, so the cells are differentiating over time. So they are, and we definitely see that they upregulate E-cadherin, for example. So it could be that as they have more E-cadherin on their surface, that is able to signal, and maybe that gives different signals that then crosstalk with the CIL signals. But yeah,
right.
that's
do you see big differences in how the conversion behavior is in those row A active versus the dominant negative?
Oh, that's a really good question. No, we haven't done sort of those really high resolution time lapses in the dominant negative row A embryos. But yeah, that's definitely something that we'd be interested in doing.
Stephanie, can I ask a question to follow Leonard's?
Sure.
Well, so I think that what Leonard's saying is very interesting, but when I looked at your data, what I think you were showing us is that when the cells make contact, actin is depolymerized at the contact site and actively polymerized opposite or nearly opposite. So if we go by Occam's razor, does that not explain the behavior in and of itself? They are going to move away as actin is polymerized on the other side of the cell.
Mhm. Yeah, so so that's a great point. So I think so what the contact is doing partially is it's orienting the cell. So before colliding with another cell, you know, the cell has a leading edge, you know, one leading edge that's in a particular spot. And so by colliding with another cell, what that is doing is it's reorienting, it's telling the cell, you know, to put your leading edge somewhere else, not, you know, not here.
So I think that explains the moving away behavior. That's all I'm saying. The second part of Leonard's point, though, was as they move away, could another program be activated that would change their behavior? I think that remains an open question. But I think the first part of it, I would be satisfied. You don't have to go beyond what you see there.
Yeah, but I guess the question is, though, what happens to allow them to converge? So yeah, so that's the million-dollar question.
Yeah, I had sort of a related question to the convergence part, because, you know, as we're discussing, it's clear that it might be that contact inhibition of locomotion stays on a little bit, even as maybe they get stickier or there's some chemotactic signal that brings them to this line at the end. And I was wondering if you thought about whether that could fix some of these issues that Tom is seeing in the model, where once they get there, they're either like highly concentrated at the poles, or in another hypothesis, they're highly concentrated in the middle. If they still have some degree of, you know, contact inhibition of locomotion, would it uniformize them along that line once they get there?
Oh, that's a really good point. Yeah.
I mean, maybe that would be a benefit to this sort of inner conflict of like, okay, I'm seeing signals from
I don't know how difficult that is to incorporate into the model.
Yeah, I guess it comes back to this idea of slowly changing, tuning contact inhibition and interactions, and yeah, finding a way to look at that.
So
It's still, I think we need to know what the signal is that they're ultimately following, like regardless of interactions. I think, you know, how do they know to head?
Yeah, because if it is a chemotactic signal, I thought to myself, wouldn't that be exactly the same as your find the closest point rule, where they would still get kind of aggregated, you know, overly populated at the poles?
I've started looking at that. Oh, go on, Eric.
Oh, sorry. I was just going to say it probably depends though on, like, because if you look at like the diffusion dinghy that he did with the line, I think that was where it's being uniformly produced, and you do get like non-trivial patterns. But I don't know if that's just because you're constraining diffusion of the molecules to the surface of the sphere, or if they could actually start diffusing. I don't know. Anyway, sorry. But all I wanted to say is that like the idea that the production of the chemotactic signal would be uniform along the lateral line that's not necessarily true, right? So that could also be something that could generate a gradient that has the structure that could uniformize the way they end up, right?
Yeah, that's exactly what I was going to say. And then with the extra point that also that diffusion across the surface of a sphere is also slightly different. I mean, if you imagined a 2D rectangle, it would be very simple, but over the surface of a sphere, actually, even if the poles are diffusing as much as the center, that molecule, that morphogen, has a larger area to cover. If you imagine, like at the equator, there's a kind of a concentration, an increase of concentration just by the geometry. So it's not, it needs the model, I think.
So speaking of geometry, I wanted to jump in and sort of go back to the data analysis. Is one thing you were talking about was how you're going to compare the predictions of the experiment to the data. And one thing that I noticed in the data where you had got the dominant negative rho A is that the overall distribution of nearest neighbor different distances wasn't that different, but the topology was different. So I was going to suggest thinking about using something like persistent homology or something like that to try to characterize, because if you imagine you have a donut, right, or a torus, the point is that if I equally space points on a donut, they're going to have the same nearest neighbor distances, approximately. That distribution will be similar compared to a sphere, right? But the point is that globally, those are topologically very different objects, right? So the point would be that maybe, because if you look at the picture, the dominant negative rho A has a big hole in it, and the other one doesn't. So maybe, I don't know if you all know anything about this persistent homology thingy. It's really simple idea. And on the surface of a sphere or anything that's roughly locally two-dimensional, it's very easy to implement. So I was just going to throw that out as a way. And the other idea that I had while I was watching what you're talking about was that your measure of uniformity is kind of cool, but it's kind of an average. It's sort of comparing what you see to what you would have with the average of throwing the things randomly, but you only have so many cells. So maybe what you really want to do is actually calculate something like a Kullback-Leibler divergence between what you observe and the uniform distribution, or some sort of information like entropic measure of the difference, and then you could compare those to what you see in the data. Anyway, I just wanted to throw that out there.
No, I completely agree that like our measure at the moment for dispersal is very basic, and I think definitely to compare with the data and with how things look in the embryo, we'll need something different. Probably this grid-based method is really just a beginning. It's too simple.
Yeah, and also one thing I also noticed, just to say, is that the formation of the aggregates when you turned off contact inhibition looked actually a bit different from what was happening, at least in the one picture we saw with the DN rho A. So maybe that's something else that you can think about as ways to sort of, because if that, I think that's kind of cool that it doesn't work as well in the model, but it may not actually sort of, I don't know, phenocopy structurally what happens when you turn off or try to turn off whatever rho A is doing in the cells. And that might be helpful. Like having a quantitative way to talk about that might be helpful.
Yeah. In the model where you see those aggregates, the persistent aggregates are really around that the equator of the sphere, and they originate right at the beginning of the model where the cells are very close, and you have a few small aggregates forming, and then they grow and persist. And I mean, we haven't looked exactly at the data, but like I don't think that's what's going to happen in if you look in the embryo. I don't think when we look at a later stage we find those aggregates bundling around the equator of the embryo. So so that there's something that needs to be changed then. But then with those, the videos I showed you were the extremes of low and a very high persistence length. So we might, somewhere in between there, we might have something more close to the data. We should check.
So there's a few questions over on the page that maybe we can address.
Sure.
So a number of them are about the migration, convergence that we've talked about, and also the influence of the contact inhibition. So I guess I'll just say really quickly to kind of address that in my mind that just as we've been talking this out, an idea that makes a ton of sense to me, like I said, is that this idea that maybe as the cells move apart, some molecular program changes and then they begin to converge, perhaps because of a chemotactic signal. But then the idea that as they're coming close together, as they're converging, contact inhibition may be getting activated again, and perhaps that helps them spread out evenly when they hit the midline or something. So perhaps it's a kind of a mixture of chemotaxis and contact inhibition all working together, right?
Mhm.
Which, you know, it's like a complex system, might make sense. So like I said, there's some questions in here, you know, would contact inhibition not be an obstacle during the convergence process? How does cell-cell adherence influence the model? And can that also account for directionality? Do the cells slow down when they approach their final destination? And is that also modeled? I think that's all part of the same kind of question. There's also was a question in the chat about whether cell division, whether you saw cells dividing in the movies.
Yes, we do see, yeah, so cells are still dividing, not super frequently. I forget exactly what the rate that we came up with was, but so,
Well, it's only a few hours, right?
Yeah.
Yeah, and I don't know how fast zebrafish cells divide, but it's not going to be many for like most, like certainly with mammalian cells, you wouldn't see a lot in that short period of time.
In terms of the model, we've done the simplest thing, which is to put the number of cells that we see in the embryo at the start, and then when we look towards the end, when we count the number of cells we see, we put in the number of divisions that we need, and there's kind of randomly, they happen at random times, because in the embryo it doesn't all happen at one time, so it seems that they're probably randomly timed.
It's some very old, cool work from tissue culture where two cells that divide take paths which are mirror images of each other.
Oh.
And you would be in a position to evaluate that.
Yeah. No, I haven't. Yeah, that would be cool to look for. Okay.
They're born with contact inhibition of locomotion.
It's not.
Yeah, I mean, this didn't get a lot of traction after it was reported, but it was very exciting at the time.
Okay, that's interesting.
Whichever way you turn, I'm going to turn the other way. Anyway, so I saw a question in the page that I thought was really cool. So I'm just going to ask it, which is that, like, is there any way experimentally to, in this embryo, look at, so you have a hypothesis for what the contact inhibition signal might be. Do you have any hypothesis for what this chemotactic signal might be, or is that known? And is there any way to actually just, because we were talking about like, what would its distribution be? But maybe there is an empirical fact about what that distribution is, if there is a chemotactic signal, and maybe you could just measure it.
Yes. So, so based on work from other labs, I think the chemotactic signal is CXCL12. So it's a chemokine. So people have knocked it down, and the endodermal cells, they don't get to the right place. So what that gradient looks like, though, people have tried this with all kinds of gradients, right? And people are still arguing about whether those are valid. So yeah, so that's an entirely different question, but yeah.
Is it a technological thing? Like
it's
hard to know how to label it or something, or?
Yeah. So so at least in zebrafish, there are very few good antibodies. So people will do things like they'll express a GFP fusion, but then there's a question of, well, if you put a honkin' GFP molecule on it, isn't that changing its diffusibility? And so, so how do you actually visualize this gradient? So, and then there's the question of like, well, even if you can visualize a gradient, is that the same as like what the cells actually sense and respond to? Because it could be that the response is what's graded and not necessarily. So it's
well, there has to be information for a response. Like that doesn't make sense as an argument unless there's other information, because the cell has no GPS locator, right? It's the only way for it to know information about where it is in some sort of coordinate system is for there to be something that it's sensing, right?
Right.
That's
Yeah.
So the point is that what that argument corresponds to is other chemotactic signals that we don't know about, which is interesting. But if you find that the distribution you observe empirically is sufficient using a model, that would be one argument that, well, maybe if it's there or not, it's not necessary, right?
Yeah. Also, I mean, I think, at least for the purposes of modeling, I don't think we need to know any of the specifics about the chemotactic signal. I think we just need to know about, so how are the cells getting to the dorsal midline? So I think if we had more information about that, so if we take more movies focused on getting that information, we can put that into the model, and then it's sort of agnostic about what the actual signal is if we just know what the cells are doing in response.
Yeah, I agree that if we could image some parts of cells and somehow analyze that in 3 dimensions so that we can really compare that to the model, it would suffice at least to distinguish between the 2 strategies I showed you there, like one video of looking at the animal pole to see if cells all go towards that animal pole would show us that that isn't the right strategy. And for the east-west strategy, you can look again at even the animal pole would be another good place to look, because you would see bizarre patterns there, basically, where they're kind of taking circles if you look down on it. So I think the model can be used there to compare paths from different strategies, and it could be useful.
At this time, I am going to, end our discussion and your wonderful project presentation, but thank you so much, Stephanie and Tom. That was fantastic. And thank you to our experts for leading us in a great discussion afterwards. A really quick agenda change before you leave. You're going to get a slightly longer lunch break. Pardon me, that's why I was late turning on. I keep coughing. If you could please be back to kick off our afternoon at 1:15. So a slightly longer break, 1:15. We would love to see you back. And again, thank you, Stephanie and Tom, for a great kickoff to our Thursday. That was great.
Thank you.
Thanks, everyone. Have a great lunch break.